Hello, everybody. Welcome back to our biology class. Today we're going to cover enzymes and DNA. Just a little heads up, enzymes is going to get quite difficult, so if you have any questions, feel free to ask. And if you don't get a chance in class or I don't answer them in class, you can always email me. So let's start. Okay, so DNA structure. So if you remember last class, we went through carbohydrates, fats, and lipids. So this class, we're just going to go through our final macromolecule, which is DNA. If you remember, macromolecule is just a polymer made up of subunits called monomers. So DNA stands for deoxyribonucleic acid. And this chemical substance is present in the nucleus of all living cells and all living organisms. And also DNA kind of determines what kind of cell is formed. And we'll get into how DNA controls that in a later lesson. Today we're just going to be talking about the structure and basic functions of DNA. And so DNA is kind of called the blueprint of life because it contains the instructions for the making of proteins within the cell. So DNA structure. So DNA is a polymer. If you remember, that's just a macromolecule made up of subunits. So the basic shape of DNA is like a ladder. And if you look here, you can see the outside of the DNA is deoxyribonucleic acid. We'll get into how that's named, and it'll make more sense. So it's just deoxyribonucleic acid. So the basic shape of DNA is a helix. So the reason that we call it a helix is because you can see this intertwining shape of like the strands winding up and down. So that's what we call the double helix. So what's the structure of DNA? So DNA is made up of nucleotides, and nucleotides are these subunits, the subunits of DNA, or the monomers. So they have three main parts that you guys have to know. So they have a simple sugar, or a 5-carbon sugar. If you remember, 5-carbon sugars are pentoses. So that's right here, all of this. That is a simple sugar. Then you have a phosphate group, and a phosphate group is PO4, and that's shown here. And then you have the nitrogenous base, which is shown here. So the way that these subunits come together is in this form, right? So you have the phosphate, you have the sugar, and you have the base. And so the phosphate of one nitrogenous base connects to the sugar of the next base. And then that chain just continues up and up and up the DNA molecule. So the way that the bases are attached is that they're attached laterally to what we call the sugar phosphate backbone. And the reason it's sugar phosphate is because if you just look at the sugar and the sugar and the base, or sugar and the phosphate, what you get is this chain. So pretty much what happens is that these bases, if you remember, that's part of the subunit, they can have variation. So as you can see here, there's a parallelogram, then there's a hexagon here, then there's a pentagon here. And as you can see, these depictions are just like diagrams. They don't actually look like hexagons and pentagons like this, but these are just depictions of that. There's many different nitrogenous bases. So there's 4 different bases you guys have to know. They're divided into pyrimidines and purines. So pyrimidines are thymine and cytosine, and purines are adenine and guanine. Thymine is always depicted with a T, cytosine with a C, adenine with an A, and guanine with a G. So the difference between pyrimidines and purines is that pyrimidines have a single ring, which you can see here. There's just one ring here. And then purines, they actually have two rings. And you can see that here. There's two rings. So what's important about these bases is that certain ones join together always. So always adenine and thymine will join together. If you remember, adenine is a purine, and then thymine is a pyrimidine. Then cytosine and guanine will always join together, guanine being the purine and cytosine being the pyrimidine. So this is important. Remember that adenine and thymine always join together, and cytosine and guanine always join together. So I want to go back to this diagram so it makes it, you guys can see. So you can see, remember we said the sugar phosphate backbone? That's here. You have the sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on both sides. But what you also have are the bases, right? So you have adenine here binding to thymine here. You have guanine here binding to cytosine here. Now, if you look up this chain, you will never see an adenine bonding with anything but thymine. Adenine always binds with thymine. So if you have an adenine here, you'll have, or if you have an adenine here, it'll bind to a thymine. That's really important. So that's like kind of the structure of DNA, but there's one more thing that you guys have to know. Okay, so it's kind of blurry, but no, we're still showing the same thing. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. And over here, we see the same thing. Phosphate, sugar, phosphate, sugar, phosphate, sugar. But what's interesting about these things is that they're flipped, right? So in this case, the phosphate is here, the sugar is here, and the base is right side up. Here, the phosphate is here, sugar is here, and the base is upside down. So that's something really important about DNA. And we call this or we call the strands antiparallel. And the strands are anti-parallel because they're parallel, because they're going in the same direction, but one is flipped. Just one is like rotated 180 degrees. And so what we can then, make from this is that we can define the ends of DNA based on where the phosphate groups are. So in the strand that's right side up, you have the 5' end here, and then on the opposite side, you have what we call the 3' end. And the reason we get 5' and 3' is if you look here, we can count the carbons of the sugar molecule. So if you remember sugars, this is a pentose, so it will have 5 sugars. So this is like a chemistry concept on how to count the sugars, but all you guys need to know is that this is 1, 2, 3, 4, 5. So whenever you're given a diagram like this, you can look at the sugar and the carbon here that's bonding to the base is carbon 1. And from there, you just count. So you go 1, 2, 3, 4, 5. And you'll get the same thing here. Another way to see it is that the 3rd carbon, or 3' carbon, is always binded to the phosphate and on one side, and then the 5' carbon is always bonded to the phosphate on the other side. So that holds true even on the antiparallel strand. You have 1st carbon here binding to the base, 3rd carbon here. It's not binded to any phosphate, but it could be. If you remember, by dehydration synthesis, the OH will go away here, and an H will go away on the other molecule in your form, and then a phosphate will come and join here. So, the 3' could be binded to a phosphate, and then the 5' is bonded to a phosphate. So that's how you can figure out which is 5' and which is 3', because if it's right side up like this, then you can count the carbon and see 1, 2, 3, 4, 5. So the highest carbon in the molecule is a 5' carbon, so it's the 5' end. Look down here, 1, 2, 3, 4, 5. So 3' carbon, we consider that the lowest carbon in a DNA molecule. That's why it's the 3' end. So then looking at the antiparallel strand, it's just flipped. Like you take the 5' end, it's going to move down here, take the 3' end, and move up here. Does everybody get how we find which is the 5' to 3' strand and which is the 3' to 5'? just to make sure, because that concept can be difficult first time. And yes, we are going to record this class, and it will be posted on our YouTube channel for those of you who want to watch it again. Okay. The YouTube channel, I will post the link on Classroom after this class. All of the lectures are there from this Alphademic, so I'll post that after class. So let's just do a quick quiz like we did last class, because just to make sure you guys know it. Some of these questions are a little bit hard, like we haven't covered them yet, and we'll cover them later. So those questions we'll just skip. So for question 1, oh, I didn't cover that, the long name for DNA. But if any of you remember it, just try to figure it out. All right, so yeah, it's definitely number 2, deoxyribonucleic acid. And the reason for this is, I didn't go back here. So here, what you see is, I believe it says, yeah, right here. So the sugar, if you remember, I said that there was a sugar in nucleotide. So in DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. So look here, DNA deoxyribonucleic acid. Again, the sugar is this part, deoxyribo, and then the second part just comes from the fact that it's a nucleic acid, so nucleic acid. And then here, the sugar is ribose, and then it's a nucleic acid. So that's how we actually got the name deoxyribonucleic acid. Okay. So, yeah, good job. So you don't have to worry about 2. 2 we haven't covered. So 3, adenine always pairs with thymine, cytosine, guanine, or ribose. Okay, good. Everyone got it. The answer is thymine. Adenine pairs with thymine. So the rungs of the DNA ladder are made of okay, so it seems like a few of you got this one wrong, but if you remember what I said, that DNA actually, so remember, we considered this the backbone of the ladder. So it wasn't 3 because the sugars and phosphates make up the backbone, but then the rungs of the ladder are made up of the base pairs. Remember, adenine, thymine, guanine, cytosine. So it kind of looks like a ladder, and then these would be the rungs. So that's how they came up with that. So it was base pair. So the DNA molecule is held together by, we didn't talk about this, but if any of you know, that's great. I will explain it now. Yeah, so good job to those of you who got it. It was the 4th choice, or hydrogen bonds. And so if we go back here, I think it was, no, it wasn't this one. Okay, so wait, that's not okay, what's this one? Yeah. So here, so the bases, adenine and thymine, and cytosine and guanine, attract each other because of hydrogen bonds. And so hydrogen bonds are generally considered weaker bonds because there's no actual physical exchanging of electrons as in covalent and ionic bonds, and they're just based off of slight charge differences between the molecules. But there's millions or trillions of base pairs in the cell, and so because of that, that actually provides a really strong seal to the DNA. It's actually one of the strongest molecules because of the amount of hydrogen bonds it has. And so another key fact is that adenine and thymine will always form 2 hydrogen bonds between each other to hold each other, and cytosine and guanine form 3 hydrogen bonds. So if you look here, this is depicting guanine, cytosine. The way I know that is because it shows that there's 3 hydrogen bonds, meaning that cytosine and guanine have to bind. Another way that you can figure out what base pair this is and what base pair this is, if you remember, we talked about purines and pyrimidines, right? Pyrimidines have 1 ring, cytosine is there, and guanine has 2 rings. So going back here, this has to be guanine because it has 2 rings, and this has to be cytosine because it has 1 ring. So that's how you guys can figure out what bases there actually are based on hydrogen bonds and things like that. So yeah, this was hydrogen bonds. 6 we didn't do, 7 we didn't do. So 8, the twisted ladder shape of DNA is called a again, good job. Most of you got this. It is the double helix. Remember that the ladders or the phosphate sugar backbones actually twirl into that helical shape. So that's why it's the double helix. So the sugar found in DNA is number 9. Remember, just think of the name. What does DNA stand for? And that should give you your answer. Okay, so the answer here was number 2, or deoxyribose. And remember, DNA is called deoxyribonucleic acid. And so it's the deoxyribose sugar is in DNA because it's called deoxyribose. That's how you would get this one. You don't have to know 10, you don't have to know 11, you know 12. Okay, so let's do 13. If you guys don't get it, then I'll just explain it again, or I can explain it. So the answer was all of these, and I don't expect you guys to know this because I didn't explain it, but I'll get into it. So remember that RNA is ribonucleic acid, which means it has a different sugar. Remember we said ribose is in RNA, deoxyribose is in DNA. Another big difference is that RNA is single-stranded. For the most part, most RNA molecules are single-stranded, and most DNA molecules are double-stranded. That's why when we talk about DNA, we talk about it having a structure of a double helix. But RNA just has a single-stranded structure, or we just consider it a strand because there's only one of them. So it's nucleotides. Remember, DNA was adenine, thymine, cytosine, and guanine. For RNA, it's adenine, uracil, cytosine, and guanine. So uracil will replace thymine in RNA. The interesting thing, though, is that adenine now bonds to uracil in RNA. So this will be important later. Just remember that uracil and thymine are like really, really similar, except for like 1 or 2 atoms or elements in the molecular structure. So for that reason, they're pretty much exactly the same. So we will get that becomes important later on. Just remember for right now that the nucleotides in RNA are adenine, uracil, cytosine, and guanine, and in DNA, they're adenine, guanine, cytosine, and thymine. So now we come back to this question. RNA differs from DNA because it does have a different sugar. It is single-stranded, and it has uracil. The answer to this one would be all of these. So 14, DNA is called the blueprint of life because okay, so everyone got it. That's good. So the answer was C. It contains the plans for building an organism. That's good. And then you do not have to know the last one, so our score is going to be horrible because we only answered like half the questions. But without further ado, let's get into enzymes now. So that was DNA and RNA. So we just did all of the macromolecules, carbohydrates, lipids, proteins, and DNA now. So enzymes. Enzymes are a type of protein. And they're what we call biological catalysts. And what a catalyst is it speeds up chemical reactions. So for this, we'll just take the chemical reaction of, I don't know, let's say breaking down glucose. So breaking down glucose, like the first major enzyme, is called hexokinase. So this enzyme, hexokinase, just an example, there's like millions of enzymes. We're just taking one for the sake of the class. And hexokinase is a biological catalyst. It increases the rate of the breakdown of glucose. So glucose gets converted to fructose in this pathway with hexokinase as the enzyme that speeds up that process. So without hexokinase, it would take a really long, well, not really long because, it'd still be pretty fast in your body, but enzymes just pretty much just, I would say like double the speed all the way up to make the speed go 10 times faster. So that's the purpose of enzymes. So remember that all enzymes are proteins and they have a specific tertiary shape. So if you remember, the tertiary structure of proteins was the folded structure. And the folded structure is like with the hydrogen bonding, disulfide bridges, hydrophobic interactions, all of those come together to form the tertiary structure of the protein. So an important thing about enzymes is that, okay, let's say hexokinase. Hexokinase can only turn glucose into fructose. It can't turn glucose into lactose. It can't turn like proteins into sugars. It can't do any of that. It can only catalyze a reaction. Catalyzing just means, speed it up. Catalyzing the reaction of turning glucose into fructose. That's the only thing hexokinase can do. So now let's get into the structure of the enzyme. So the enzyme, which is the catalyst, contains a structure called the active site. So in here in the schematic diagram, you can see that this is the substrate, which in our case is glucose, and this is the enzyme or hexokinase. So what's going to happen is this glucose is going to bind to the active site of the enzyme. When it binds to the active site of the enzyme, it kind of gets like bent in some sense, kind of getting the bonds in the glucose ready to break them. So because it binds like really, really tightly, it starts to break the bonds within that glucose molecule. And then what this does is we'll talk about how that actually helps, but pretty much the rest of the enzyme is like giant and the active site is like really tiny. So the active site is usually like 20 to maybe 100 amino acids, and the protein can be, upwards of 1,000 amino acids. And the reason for this is because the rest of the protein is involved in maintaining that shape of the enzyme. Because enzymes are also because they're really, really specific, if their shape changes like a little bit just by a single molecule or single bond, they won't be able to function. So that's why it's really, really key that this entire, giant structure is there, because if the active site, even if like one atom is not there, it won't function properly. So when an actual reaction occurs, the substrate binds to the active site, the substrate being the initial thing that you have. In our reaction, it's glucose. And so that glucose is turned into the product, which is fructose in our example. The substrate can be this is wrong, but the substrate can only be one molecule. Remember that enzymes are super specific. So if, let's say, a lactose molecule tries to bind to hexokinase, it won't be able to. But glucose can bind perfectly into this hexokinase enzyme, but lactose can't. So enzymes are really, really specific. So some examples of enzymes are lactase, which breaks down lactose into glucose and galactose, catalase, glycogen synthase, or ATPase. Notice how every enzyme, like the last three letters, are ACE. Anything that ends with ASE is usually an enzyme. So in the example I gave, I said hexokinase. Remember, ACE at the end, suggesting it's an enzyme. So that's like the structure of enzymes and how they work. So, or not how they work, just the structure. So enzymes are really affected by their environment. Small changes in their environment can lead to their denaturation. If you remember from our proteins class, denaturation just means the protein's structure changes, and this leads to its function being impaired. So, for example, temperature is one thing that can denature an enzyme. So, for example, we maintain our body temperature because we are endotherms or endothermic animals, and we try to keep our temperature at a constant temperature. And so this is really important, and the reason why we do this is to make sure that our enzymes are functioning properly. And that's why a fever of 105 can kill you, because it's going to start denaturing your enzymes. And a temperature of, or really, really low temperature can also kill you because, again, it's denaturing your enzymes. The same thing is with pH. So remember, pH is a measure of acidity. How many H plus hydrogen ions there are in the solution. So for example, in your stomach, right? The stomach has that gastric acid, which comes in at a pH of around 2 or super, super acidic. So the enzyme in there called pepsin, it actually functions best at a pH of 2. So, but an enzyme like hexokinase, which is in like your muscle cells, converting glucose into fructose, if you moved that into your stomach, it would immediately denature because of the extreme pH. So again, that's why we also try to maintain the pH of our cells. So how do enzymes work? So enzymes or reactions require a really, really high energy. So, and this is what we call the activation energy. I'm just going to show you an image of what this would actually look like. So just in this diagram here, this is what we call an energy diagram. So right here, you can see that the energy is on the y-axis, and then the time is on the x-axis. So the energy required to start the reaction is called the activation energy. So if I wanted to turn glucose into fructose, I would need a certain amount of energy to do so. If I don't have that energy, then the reaction can't occur. That's what's shown here. So the blue curve here is the activation energy with no enzyme. So if I have no enzyme, I would need this much energy. I would need to take the reactant and add this much energy, which is called the activation energy. And then the reaction will occur and the energy, comes down because we're breaking down that molecule or something. But the reason enzymes work so well is because they decrease the energy needed to start the reaction. So look here, the actual activation energy is only this much. It's much less than this activation energy. So they decrease the amount of energy required to actually start the reaction. So coming back here, the way that they do this is they put stress on the bonds within a molecule, or the molecules that are close together. And remember I said it kind of bends those bonds, and because it puts stress on this, that's almost like the enzyme uses energy and starts to bend those bonds. And because of the bending of the bonds, it decreases the activation energy. Now, another interesting thing is that temperature, or we said that very high temperatures can denature an enzyme, but temperature. This will be important too. So here, so you can see again in this graph that the temperature is at the bottom and the activities of the enzyme is here. So, right, this is like that point where our body just, all the enzymes kind of denature because, there's no enzyme activity past this temperature. But if you actually look at it, the peak enzymatic activity is around up here, which is around like 41 degrees Celsius. Our body temperature is around 37 degrees Celsius. So why is this graph saying that the peak is around 41 if, if our body temperature is around 37? Well, the interesting thing is that, a higher temperature actually makes enzymes go faster or work faster, because higher temperature just means more energy, and more energy means, again, the activation energy of the reaction is decreased. So, in our body, when you get sick or you have the flu or something like that, one of the first symptoms that you have is a fever. And the reason fever works is it's your immune system, heating your body so that your enzymes work faster to kill the pathogen or whatever is infecting you. So that's how a fever works. It just increases the speed of your enzymes. So the reason that your body is at like 37 degrees Celsius and not 41 is because, so there's two reasons. One is that different enzymes work best at different temperatures. So some enzymes may actually work best at 27 degrees Celsius. Some may work best at 100 or like 100 degrees Celsius, who knows? So the average temperature is what is shown in this graph. This is like the average temperature of all enzymes. So that is really important for immunology because it relates to how well your enzymes function. So that is the effect of temperature on enzymes. So now let's go into, a little bit more advanced, two hypotheses, lock and key hypothesis and the induced fit hypothesis, on how the enzyme and the substrate really bind in the active site. So the enzyme and the substrate in the lock and key hypothesis fit, like a lock and key. The substrate fits perfectly into the enzyme. But this is actually flawed, and no enzyme actually uses this model. And the reason for that is quite advanced, but all you guys need to know is that the lock and key hypothesis is invalid, and it's been disproved because this never happens. What actually happens is what we call the induced fit hypothesis. So as you can see here, the enzyme does not match what the substrate looks like. But pretty much what happens is after the substrate comes in close proximity to the enzyme changes its structure to better fit the substrate. So that's what the induced fit hypothesis is. So those are the two models, the induced fit and lock and key. Remember that lock and key is outdated. We don't use it anymore. Enzymes do not perfectly match their substrate. We use the induced fit being the enzymes come together with the substrates, and then the enzyme changes its conformation or its structure to better fit the enzyme. So that is what we're going to be doing for enzymes today. I do have a quiz, but our time is going to run out. So if you guys could log back in after I shut down this meeting so that we can do the quiz, that would be great. So just quickly log back off and then log back in. Okay, so let's start then. One, without the presence of enzymes, the reactions necessary to sustain life would require blank. Yeah, so the answer here was 2 or higher temperatures. And if you remember, we said that higher temperatures mean faster reactions. So if we didn't have enzymes, our body would probably be at higher temperatures. So good. Question 2, we didn't talk about, so I will just explain what it's showing now. So if we graph the substrate concentration, remember the substrate being this initial stuff, and then the reaction rate on the y-axis, where the reaction rate is, how fast these enzymes are converting the substrate, then what we get is this curve, which is called a hyperbola. And the reason it's called a hyperbola is because by A, or at this point, this is where the enzymes are working at their maximum, what should I call it? Like their maximum capacity, right? Like they're working really, really hard. Well, actually, no, this is substrate. No, sorry, scratch that. At this point, there is not that much substrate, right? So if there isn't that much substrate, the reaction rate is going to be low. But what we see later and later on is that, eventually the reaction rate kind of like cuts off. And the reason it cuts off is because the enzymes are what we call saturated. So when an enzyme becomes saturated, it means it's considered full of substrate. Like it can't bind to any more substrate, it's full. So the reason we get to this point is because all of the enzymes that we have become saturated. And so even if we increase the substrate concentration to infinity, the reaction rate would still stay the same. So yeah, so now think about question 2. Based on the graph, which of the following could be used to increase the reaction rate beyond point C? Okay, so it seems like this was a more difficult question, but the 2 who answered, good job, you both got it. So think about this, right? If we increase the amount of substrate, remember I said that that's going to have no effect. Increasing the amount of substrate does not change anything in the reaction. The reaction rate is still at its maximum. Adding more water, enzymes pretty much have nothing to do with water, and decrease the enzyme concentration. If we decrease the enzyme concentration, we would actually decrease C. And the reason for that is because with less enzymes, the reaction rate can't go as fast. That's like decreasing the temperature of a reaction because you're just taking away energy from the reaction in the form of enzyme. So the best answer here is increase the temperature, because increasing the temperature, again, it just provides energy for the reaction to occur. So the part of the enzyme where the substrate binds is called the So the answer here was active site, or 1. Remember that catalyst is the enzyme itself. Catalyst is another term to describe a biological enzyme. Inhibitor, we haven't talked about yet, and large subunit, we haven't talked about yet. Four, when a piece of liver is dropped into hydrogen peroxide, you guys don't have to know this. This is talking about a specific enzymatic reaction. So we can do five, though. Some people cannot digest milk products because they lack a specific enzyme. Which enzyme would be used to break down the lactose in milk? So, good job to the people who got it. It was four. It was lactase, and you can pretty much derive the name of the enzyme based on its function. So, lactase breaks down lactose. Another enzyme called DNase, DNASE, breaks down DNA, and then there's RNase, which breaks down RNA. So anything that ends with ase and has the same like suffix or prefix, you can use that to figure out what that enzyme actually does. So 6 on the image, which letter represents the enzyme? A, B, C, or D? I had marked these answers. So the answer was actually B. So for those of you who got it, that's good. So the reason that it's B is because remember that you have the enzyme with the active site, and you can see that there's things going into that active site and things coming out. So that's how you would know it was the enzyme. So which letter represents the substrate? A, B, C, or D? So for this one, it was A. And so pretty much everybody got it. So for those who did, good job. So remember that substrate is the initial step of the reaction. So those are going to be going into the enzyme. Remember the substrate and the enzyme bind together through the induced fit model. So that's going to be the substrate. Now, which is the product of this reaction? Again, A, B, C, or D? Again, everybody got it, so good job. So the answer was C. And the reason for this is because the products is what comes out of the enzyme or what comes out of the reaction. So here, in what way could you increase the rate of reaction as in the image of C? So this was a little bit of a tricky one, and this actually has to do with the curve here. So if you remember that at point C, the enzymes are saturated, and that's actually what's being shown here. You can see that all of the enzymes are bound with their substrate. So like this is the red is the substrate, the large orange thing is the enzyme. And you can see in A and B that it's not happening. They're not fully saturated. So in C, they're saturated. So the only way we could increase the reaction rate is adding enzymes. If we add more substrate, remember that on this graph, it doesn't change the reaction rate. It still stays at its maximum. So finally, enzymes are composed of what organic molecule? Sugars, DNA, fatty acids, or protein? So yeah, good job to everyone who got it. Remember, they're proteins, And remember that for this reason, they have the properties of proteins, like they denature with high temperatures or weird pHs. And if you change, and they have a primary, secondary, tertiary structure. Some may even have a quaternary structure. So enzymes are proteins. Okay, so good job. We got all of them right that we answered. So that's it for today's class. We learned about enzymes and nucleic acids. Next class, we're going to finish up a little bit more of enzymes. There's like a few more details that I want to cover, specifically about like reactions and things like that. And from then, we'll start doing cell and cell biology. So if you guys have any questions, feel free to ask them now. If not, then we will be done. Okay, so that's nobody. Then thank you all for attending. I hope you learned something, and I will see you next week.